Hello and welcome to the FDA Science Forum session on food and cosmetic safety, the role of innovation and technology. My name is Chad Nelson and I am with FDA Center for Food Safety and Applied Nutrition. I have the distinct honor and privilege of introducing the distinguished speakers for today's program. First, we will hear from Dr. Callie Neal, Professor of Animal and Food Sciences at the University of Delaware, One Health as a collaborative response to food safety risks. Our second speaker will be Dr. Susan Main, the Director of FDA's Center for Food Safety and Applied Nutrition, who will present on CFSAN's use of innovative science to address current and emerging public health priorities. Next up, we will have Dr. Dan Rice, the Associate Director of the Office of Food and Feed Laboratory Operations in FDA's Office of Regulatory Affairs, who will talk about FDA's support of recent foodborne illness outbreak investigations. Our fourth speaker will be Dr. David Edwards, Director of the Division of Animal Feeds and FDA's Center for Veterinary Medicine, who will present on what won't an animal eat, innovation in animal diets. And last but not least will be Dr. Luisa Camacho, a research biologist in the Division of Biochemical Toxicology at FDA's National Center for Toxicological Research, whose presentation is entitled Mind the Data Gap, Contributions of FDA's NCTR to Evaluate Cosmetic Safety. Following the presentations, Dr. Jeff Ward, Senior Science Advisor in FDA's Center for Veterinary Medicine, will moderate our panel discussion. I encourage you to visit FDA Science Forum's website and read the full bios of the speakers in the forum brochure. Now I'd like to invite Dr. Neal to turn her camera on and begin her presentation. Thank you, and I hope you enjoy our session. Dr. Neal?
Good afternoon, Chad. Good afternoon, everyone. Thank you very much for the opportunity to be here with you today. It's really quite an honor and a privilege. And I'm very excited to tell you about One Health as a collaborative response to food safety risks. This is the University of Delaware campus for agriculture and natural resources, my college. And I wanted to show you this picture because it really does showcase One Health. We have a 300-acre laboratory right outside our doors. And we have greenhouses for the plant scientists as well as food scientists. We have dairy and beef and sheep, and we have students everywhere, right? So this is really a wonderful showcase for how we think about collaborations within a One Health context. And many of you are probably familiar with One Health, and I'm going to tell you a little bit about how I see One Health in terms of making connections. We really are looking for the optimum health of humans, animals, and the environment. But I want us to think more about how we get to that optimum health, the connections that we impact in doing that. I think that's what I really want to tease out over the next few minutes. And a little bit of background. One Health is a historical concept. It's not brand new, but many people are using it. It did start in 400 BC with Hippocrates on airs, waters, and places, and maybe some of you are familiar with that. It has been used in referring to zoonosis, and those linkages were described by Rudolf Virchow, who was a prominent German physician in the 19th century. In addition to his medical career, he really advocated for the importance of improved veterinary education. And that's really where One Health has been for a lot of the most recent years in particular with veterinary schools. And the UC Davis Veterinary School coined the term One Health, using it as a way to think about collaborations to effectively cure, prevent, and control illnesses that affect humans and animals. And then you can see we've even broadened that scope a little bit. The CDC established an Office of One Health in 2009 and the USDA in 2012. Of course, many of you are familiar with this growth in population and our need to find a way to better advance food security and food safety. One Health provides a way to be able to ensure the well-being of people, animals, and the environment. As we think about creative and collaborative problem solving, we really need to think outside of the box because we have limited resources, and so we need to work together. We cannot continue to feed people or animals in the way we are if we do not think about the impact on the environment. And as we are thinking about this connection between animals and humans, I would be remiss if I did not mention, at least briefly, what has been impacting our world for quite some time now, and feels like a long period of time, I think to many of us, the origin of SARS-CoV-2 virus is still something that is being considered by many in different ways. But what we know about coronaviruses is that they frequent various animal types and animal populations. There are multiple spillover events that are possible. There are new spillover events right now that are being discussed by scientists around the world. And it's important that we understand that these spillover events may happen, and we need to think about how we're interacting and how we're surveying the environment. Really, that's that collaboration between these. Unique aspects of One Health have occurred over the past 15 months or so, and this is showing a couple of those thoughts about One Health. I attended a meeting by landscape architects where they spoke about the embracing a One Health concept for how we are designing new outdoor spaces, because these have become even more important to us over the past year. Here's an image up top from New York, and you can see the creative use of the outdoor space. And the slide image down there is one that I took a screenshot of during this meeting, where landscape architects were talking about One Health concepts in jungle environments, where some of these coronavirus spillover events or other viruses may be surviving and intermingling with multiple species of animals as well as humans. And so we can really broaden One Health to not just the veterinary schools anymore or the medical schools, but really to so many different contexts. And that's where I hope we continue to have this collaborative thought. Right? And I love this infographic from the CDC. If I skip down here, to achieve the best health outcomes for people, animals, plants, and our environment, how do we get there? We have to have coordinating communication and collaboration. And without that, we are never going to achieve that. So we tend to stick in our silos. We tend to want to really address detailed questions of science, but we must approach these with a multidisciplinary and transdisciplinary approach in order to be able to achieve this optimum health outcome for all of us. And this is not just an issue in the United States. There are international collaborations on One Health. Here's some information from the OIE, and you can see here they are speaking directly to some of the zoonotic diseases of importance in both animals and humans. And many of the existing human infectious diseases are zoonotic, about 60%. Emerging infectious diseases are very high to be zoonotic disease, as we've seen with coronavirus. And so again, we have to think about how those interactions are happening and how we think about those collaborations to make health optimum. As a food scientist and food safety specialist, this is an image that I use in some of my classes, and you have perhaps seen this as well. And if you may be able to use the QR code there, otherwise you'll have access to this video. But the well-traveled salad is a wonderful representation of how we are One Health collaborative community when it comes to food production. We need to think about how we are producing food, transporting food, distributing food, when we think about food production and food security, and making sure that we all have access to those delicious food products such as those shown here. And I know it's right after lunch or right at lunch, and so maybe some of you are enjoying this type of a global food product. So no matter what your role in science is, I believe that most of us, if not all, can have a role in terms of a One Health context. When I think about food safety, it really embraces One Health, and in fact, it may actually be the epitome of One Health. We must have adequate food and water supply to prevent disease. We must have that to be able to produce feed and food for animals and humans. And as a reminder, in the United States, about 1 in 6 Americans get sick each year from something he or she ate. So there is an important takeaway from this, in that we want to think about how we are producing food and who is getting what food to make sure that we can really, so we can make sure that we are connecting this collaborative aspect between humans and the environment where it comes to growing our food. And in fact, humans are really important because we play many roles in this, not only as scientists, but also as consumers. And when we think about emerging infectious disease in the context of ecosystems, that's also an important role when it comes to what consumers are asking of how we produce our food. And I think that consumers are really driving some of the change in ecosystems when it comes to food production. For example, more and more consumers want to eat local, which is a wonderful opportunity. It's a wonderful choice that we have, and in fact, something that you see and we all see across our food production supply chain are more and more choices. But consumers are really advocating for these choices, and they are driving change in the food production systems are responding to what consumers want. Some of that is not necessarily embracing the One Health concept, right? When we think about how important the optimum health is, we moved to production of animals in terms of indoor facilities, and you see here with swine and poultry, we moved to these types of environments because we wanted to be able to maintain quality, safety, and health of the animal. When consumers would like animals to be in a happier environment, to be outside and have access to the outdoors, that is not necessarily better for the health of the animal or the health of the human who will be eating that animal. In fact, we see an increase in chance of protozoan pathogens and various parasites, helminth parasites as well, and sometimes there's increased mortality. And we're also removing antibiotics from feed, which is not a bad thing, but we really need to think about what challenges come after we remove antibiotics from feed. And not just feed that was used here in poultry for growth promotants, because we've removed that for quite some time, but when we think about the health of the animals, how do we respond to that and what happens to the environment? And so there's a lot more at play here than just making consumers happy and make them feel good about the food that they're getting. Of course, that's important as well. The intersection of One Health and food safety can include multiple issues, and some may be more familiar than others, and this can actually be quite complex interactions. If we look at these three different outbreaks that are all associated with Shiga-toxigenic E. coli, you can see here that we have a bison, ground bison, we have a plant product, and then we have an unknown source, right? And so they're all caused by the same organism, but very complicated issues that are very different surrounding each one. And certainly our pets are important animals to think about when it comes to One Health as well. And this is a showcase of various outbreaks, multi-state outbreaks on the CDC website that happened over the past few years that all involve salmonella for the most part. Yes, all salmonella, or few of them have hantavirus or campylobacter that were involved in multiple types of pets. And so that's really important. One of the larger growing populations of pets are backyard flocks. And even during COVID, we really saw an increase in individuals who wanted to have their own food production. And so we see this increase in the growing of backyard poultry. This was an outbreak that is now concluded, although there is a current outbreak going on of salmonella linked to backyard poultry. And so when we and that is, has about 163 illnesses as of today across 43 states and 34 hospitalizations involved in the one today. That's on the CDC website if you'd like to know more. And so we really want to think about what it is about salmonella that makes it such a good pathogen when it comes to One Health. One of the issues is that the transmission occurs here between humans, animals, and plants, and salmonella can survive very well in those different environments. We also have seen multiple outbreaks occur with poultry, with fruits and nuts, with vine stock vegetables, and salmonella is able to survive in all of these environments. This paper here really showcases how salmonella is able to survive across multiple host species and in the environment, and does that in a much better way compared to E. coli. And what's interesting is that we've seen changes in terms of salmonella, right? We've really seen a decrease in the animal salmonella that we were more familiar with, Salmonella pylori, and it has been replaced with cases of salmonella in humans. So there's a lot that has to do with the way we interact with animals, the way we interact with our environment. And I can show you just some quick data of a paper that we have that goes into these differences. If we inoculated a salmonella cocktail here in different species from different animals, I just want you to take a look at this, and you can see there is such a difference here of the survival of fecal material from different animals across different states, right? And so you can really see those differences here. And if you look at what salmonella survived, each salmonella acted individually, very different across the others. So there's a huge amount of information to dive into salmonella. So in the last few minutes that I have, I want to move away from the bacteria and talk a little bit more about a virus that is also a really wonderful example when it comes to talking about One Health, and that is avian influenza. You may have seen this infographic here because as we talk about RNA viruses, the novel coronavirus changes more slowly compared to influenza virus that we know mutates very well and adapts extremely well to multiple hosts. And you can see that here on the right-hand side. I also want us to keep in mind this growing backyard flock, right? And this is a newspaper article from the New York Times posted recently that has this lovely family embracing their new flock. And we really want to think about how do we educate people and how do we encourage this type of food production and embracing animals, but how do we also get people to think about those collaborative approaches to One Health? And influenza is a great example, as I said, of One Health when we think about how it affects humans as well as animals. There are multiple different strains that can be identified around the world, and we're going to look at just a few of those here. Of course, those mutations occur in both the hemagglutinin and neuraminidase proteins on the outside. There's both larger and smaller recombination events that happen, and those happen here in all of these different hosts. And you may be very familiar with swine flu. You may be less familiar with the role that terns or marine mammals play, but they're all very important reservoirs when it comes to avian influenza. And a couple of examples here. We have adaptation of avian flu, where we have seen this adaptation happen and occur in leading to outbreaks in swine as well as poultry. More recently, H5, H7, H9 infections directly from transmission from poultry, and that's again why we really want to think about how do we communicate to individuals when they have these backyard flocks. In the 2015 AI outbreak, we saw outbreaks in the commercial as well as backyard flocks, right? So I want to point that part out here. So backyard flocks are not necessarily always safe. And this was a huge outbreak because it really resulted in a massive loss of poultry inventory around the country. Another thing to think about is what's happening in our environments in terms of live bird markets and movement of birds. That's been a known issue. And in my university, we are very involved in terms of avian influenza because we have a huge chicken population in the Delmarva area, and it's very important economically, and so we want to make sure that the birds are protected as well as the humans, as well as the environment. And so all of that's really important, not only from a disease standpoint, but also from the economics. And you can see that here on the right-hand side, this is the Delmarva Peninsula. These are all individual family farms, and so it's really wonderful to be able to see people embracing agriculture that way. Again, this is an infographic from the CDC that you may wish to share if you know people who have backyard flocks. We really need to communicate about contact with animals, washing your hands, right, being very careful so that we can reduce transmission when we can. The last couple things I want to talk about is focus back to the environment. When we think about how is avian influenza being transmitted, right, this is a global issue. And all of these, the green, blue, red, black, all of these are flyways from different birds. And you can see they move across continents around the world in some cases. And we have these migratory flyways here in the U.S., but these are each important when it comes to movement of potential diseases as well. There have been some great connections, right, these collaborations in terms of One Health when thinking about ducks that are grazing and where are they grazing in these intensive rice production paddies, right? And so those are really important to think about where we find animals and where we are producing food. Sometimes it's impossible to disconnect the two, but in other cases, we might be able to have an impact. So if we look at what's happening in the United States, we have overlap with the lower latitude migratory wild ducks in terms of where we have a lot of poultry houses. And so we won't be able to move the poultry houses that are there now, but when we're thinking about production of food and industrial animal production, how might we consider the environment and what's happening in terms of the animals? This is a great example of that, showing that viruses were detected in both poultry and wild birds, simultaneously detection, across areas of the world here, right? We have this global connection when it comes to One Health and looking at those flyway patterns. Also, when we think about that global perspective, this is a recent data that comes from the OIE. And you can see on the left-hand side, these are actually outbreaks happening in poultry that were recently identified. And here the red dots are ones that are the outbreaks, the new outbreaks, and then the blue dots are what's called ongoing outbreaks. And if we look at the right-hand side and you kind of match those up, this is where they were detecting those high-pass avian influenza strains in wild birds. So again, there's overlap there. And what can we do to try to understand that, to try to facilitate collaboration among scientists and understanding what's going on in the environment? I want to share this with you, and of course, some of these slides have come from two great colleagues at University of Delaware, Jack Gelb and Chris Williams, and this shows you what some of that effort, that collaborative effort that's ongoing in the United States. Here at the USDA, the USGS, and multiple other agencies in our government have been doing quite a bit of surveillance in the past, really looking for those high-path AI strains so that we can understand what's happening in the environment and help to educate what's happening within poultry production. And that's really important, those collaborations there, including some surveillance. Of course, surveillance in animals has come up when it comes to coronavirus, and so we really want to make sure we continue to have funding and continue to have research that's ongoing for those types of surveillance activities and monitoring what's going on in the environment. So in summary, I hope that we all continue to keep making connections, really thinking about ways to advance food safety and public health. I think it's critical for us to think about how our health is impacted by animals, not only by the pets that we love and embrace and the animals that we use domestically for food production, but also thinking about what happens with those wild animals that are around us that we enjoy if you're bird watching or outside, those wildlife that may be carrying different diseases. How do we interact with them in the environment? As we are, as humans, are really taking over more and more of those spaces, this is becoming even more critical. What can we do as scientists and educators to try to protect those environments? How can we communicate that to individuals, to our students, to consumers, so that they can all understand this? And with that, I thank you very much. And my time is up. So thank you for the opportunity to be here with you.
Thank you, Dr. Neal. Dr. Main, can you please turn your camera on? And you may begin your presentation. Thank you.
Great. So good afternoon, and I did want to thank the first speaker for really setting the stage so nicely for the perspective about One Health, and I know we'll come back to that throughout this panel session today. So welcome to the 2021 FDA Science Forum Food and Chemical Safety Session. I'm going to be speaking on behalf of FDA, and the FDA is responsible for 77% of all food consumed in the United States. And we regulate $729 billion worth of domestic food, $124 billion worth of imported food, and over $80 billion worth of cosmetics that are sold across state lines. FDA's regulatory authority over the food supply includes human dietary supplements and botanicals. The employees of CFSAN, CVM, and ORA run the gamut of chemists, toxicologists, microbiologists, physicians, veterinarians, lawyers, nutritional scientists, epidemiologists, social scientists, economists, and others conducting regulatory science-related research. That research is aimed at increasing our understanding of the underlying factors and variables that can contribute to or pose a risk to human safety and health for CFSAN-regulated products. We are a science-based regulatory public health agency, so science is really at the foundation of what we do. You will hear many talks on these areas, so I will give a taste, no pun intended, of some of the innovative science that we are using within CFSAN to address current and emerging public health threats. Next slide, please. So again, just extend the warmest welcome. Let's jump on to the next slide, please. So in terms of innovative science, traditional scientific methods are necessary to stay abreast of current techniques, products, and production. But we live in an ever-changing world, and the consumer drive for novel products, such as cell-cultured foods or edible insects, which include cicadas, by the way, and more information on the production and ingredients of a product requires CFSAN to engage in and encourage the development of new technologies for the detection, reduction, inactivation, or elimination of hazards from FDA-regulated products. Some of the areas where innovation is happening are summarized on this particular slide. So we are using predictive toxicology to inform better decisions while emphasizing our commitment to reduce or eliminate the use of animals in toxicity testing. We do consumer studies, for example, in nutrition and labeling, but also in food safety, to recognize consumer views and understanding in order to provide information that will empower consumers to make well-informed decisions. You're going to hear about some of our work in genomics and metagenomics and bioinformatics to help us clarify the root cause of foodborne outbreaks, support those investigations, and prevent future outbreaks. And on the chemistry side, we continue to develop more rapid analytical methods to set the global standards for measuring chemicals of concern. And that would include chemicals such as the cannabidiols or CBD, the PFAS substances, the per and polyfluoroalkyl substances, metals, and we also do risk assessments to inform policy decisions aimed at reducing consumer exposure to potential hazards in the products that we regulate, including food. So I'd like to give you just a brief introduction to each of these as just a few examples about some of the innovative science that we are engaged in CFSAN. Next slide, please.
Dr. Mann, can you please check your camera to see that it's on? Thank you.
Let's get back. It was on. It got me kicked out. Let me get
dropped out
back.
momentarily. Here you come.
I'm coming back in, Chad.
Great, thank you. There we go. Thank you.
There we go. There we go. Next slide, please. Okay, on to predictive toxicology. Some examples about predictive toxicology and the importance of it I'll highlight here. First, predictive toxicology can be used as an alternative to traditional in vitro or in vivo methods and animal testing, and CFSAN has been strongly engaged in this area. Currently, we are publishing multiple papers on the use of a predictive toxicology tool called the Expanded Decision Tree. The Expanded Decision Tree classifies compounds according to their chronic oral toxicity using a sequence of structure-based questions to which the answer either refers the user to another question or assigns the substance to one of 6 classes of toxic potential. The combined Expanded Decision Tree and Threshold of Toxicological Concern approach is suitable for screening, prioritization, and safety evaluation of substances with low exposure scenarios, along with the safety evaluation of mixtures. This is so important given that much of CFSAN's work is in the post-market setting, although the Expanded Decision Tree can also help with pre-market reviews. Another exciting area is developing alternative methods to animal testing for cosmetics. Look for 3 posters describing the development of new methods and the establishment of already validated methods to identify potential skin sensitizers. The new alternative methods combine predictive computer modeling and in vitro test methods to minimize animal testing. Legislation has considered banning the use of animals for testing cosmetic ingredients, so progress in this area is critically needed. And so please look for posters on both of these topics. Next slide, please. On to consumer studies. CIFSEN has a well-developed program in consumer studies providing robust research to understand consumer thoughts and views so that we can provide them understandable information, empowering consumers to make informed decisions. For example, we use focus groups to understand consumer attitudes and to determine the most effective venues, formats, and content to ensure our communications are accessible and understandable for consumers. Another application is through experimentation of how consumers understand claims and how this information can result in erroneous beliefs or confusion. These experiments provide empirical data to help support agency actions on product labeling. A third aspect of surveys is focusing in on consumers' knowledge and beliefs that offers important information to policymakers, educators, and communicators about the need for targeted public information to address knowledge gaps and better inform consumer decisions. Look for Amy Lando's presentation on the results from the 2019 Food Safety and Nutrition Survey. There are also posters on a child obesity focus group study and on data from a raw flour analysis that we did showing that most consumers are unaware of the risk from consuming raw or uncooked flour. Next slide, please. Switching into genomics, metagenomics, and bioinformatics, we've been using tools like DNA barcoding, which has been utilized to identify sequence data specific to an organism distinct from another species. For example, we have used DNA barcoding to identify seafood species. And we are taking this technique further by applying it to dietary supplements and botanicals. This will be used to test the authenticity of labeling and to prevent economic adulteration of these products. Another area of genomics, metagenomics, and bioinformatics that we are well known for is Genome Tracker. The Genome Tracker Network is the first distributed network of laboratories to utilize whole genome sequencing for pathogen identification. It consists of public health and university laboratories that collect and share genomic and geographic data from foodborne pathogens. The data, which are housed in public databases at the National Center for Biotechnology Information, or NCBI, that data can be accessed by researchers and public health officials for real-time comparison and analysis that can be used to speed foodborne illness outbreak investigations and reduce foodborne illnesses and deaths. The human and economic impact of predictive identification or rapid identification of disease and thus outbreak prevention or disruption is tremendous. And you can see posters that can be found on both the Genome Tracker Network updates and the economic impact as well. Next slide, please. So talking more about methods, an important role that we have is to help to develop rapid analytical methods. And as I just mentioned with regard to Genome Tracker and whole genome sequencing, methods that are robust, accurate, efficient, and rapid can save lives and can help improve public health. And we are currently focusing on a variety of such analytical methods. With the increased marketing of cannabinoids and hemp products, we needed to produce methods to analyze CBDs, toxic elements, pesticides, and microbial contamination in a variety of products, including beverages, food, and pet products, and powders and oils. We are currently sampling over 200 products, and we plan to sample up to 3,000 products long-term. Another area of concern is the mixture of e-waste or electronic waste in the recycling stream, which can contaminate food contact materials, for example, plastic being recycled for food packaging. The primary concern is that heavy metals, flame retardants, and polymer degradants that are not authorized for food contact could make their way into recycled materials that are being used for food contact. Concerns of misidentification when utilizing our current methods pushed us to develop and test a rapid screening method we are currently using for a survey of marketplace samples. Next, there's great interest in metals in food, such as arsenic in infant rice cereal and apple juice, and we have continued creating methods that are rapid, specialized, and lower cost. Cost is important both for our own testing as well as to incentivize greater testing of foods by industry. For example, we have adapted an off-the-shelf well water testing kit for testing infant rice cereal. By producing validated methods, field investigators can test products quicker and more efficiently. Another new area is that of portable screening devices. Consumers now have access to marketed devices that use smartphone apps or other technology that are easy to use and user-friendly. However, these tend to have low functionality and spectral range and need testing. We plan to develop screening methods that employ such portable technology. Look for the poster on the development of non-target screening methods based on vibrational spectroscopy instrumentation and chemometrics for applications such as milk powder authentication, seafood decomposition analysis, and determination of plasticizers in tubing. Next slide, please. As I mentioned earlier, CFSAN is also responsible for cosmetics and cosmetic safety. And I would encourage you to look for several posters that we have available on cosmetic safety. One poster provides an overview of new methods developed to determine the mercury levels in certifiable color additives. CFSAN certifies color additives, and these are additives that are used to color foods, drugs, cosmetics, medical devices, and in some cases, the human body. The FDA pre-approves color additives, and mercury is a known impurity. Next slide, please. Hopefully, as you can see, CFSAN continues to test industry and consumer technology while developing our own to meet consumer demands, create a safe product marketplace, limit animal testing, and provide information to empower consumers. We look forward to continuing to serve you and the country, bringing our best science to food and cosmetic safety. Thank you for joining us, and enjoy the session.
Thank you, Dr. May. Dr. Rice, you can turn your camera on. And you can begin your presentation. Thank you.
Well, hello everyone. I'm honored and thrilled to be part of this session of the FDA Science Forum and speaking on a panel of such excellent scientists. Today, I'm going to be presenting on FDA support of recent foodborne illness outbreak investigations, and I think what I'll be presenting today ties in really well with Drs. Neal and Main's presentations earlier on both on One Health and some of the technological advancements we've made to help support outbreak investigations. There are a lot of players in the prevention, detection, and response of foodborne illness. It's very much a team approach spanning several agencies at the federal and state and local levels. And so in my brief presentation today, I'll be highlighting the FDA contributions and recent investigation efforts, and also emphasizing how these other partners are so important in the investigations that we participate in. So, as I said, it's a collaborative approach. The FDA is a partner in response to outbreak of foodborne illness for most human and animal food products, the products that we regulate. And it's a collective effort spanning the Office of Food Policy and Response, the Centers for Food Safety and Applied Nutrition, where Dr. Main is director, the Center for Veterinary Medicine, FDA CORE, which is Coordinated Outbreak Response and Evaluation, the Office of Regulatory Affairs, where I work, compliance programs, inspection programs, and laboratories. And actually, I'm a laboratory person. I direct the Office of Human Animal Food Laboratory Operations for ORA, Office of Regulatory Affairs. A little bit about FDA's Coordinated Outbreak Response Evaluation Group. They manage response, surveillance, and post-response activities, and they're a group that we've, in my shop with laboratory operations, we interact with staff and core pretty much on a daily basis. In the Office of Regulatory Affairs, we have inspectional laboratory testing support. So there's the Office of Human Animal Food Operations that oversees domestic food that's in commerce in the USA. And they have staff who perform inspections of food manufacturing facilities, food distribution centers. They are the staff who collect samples that come to the laboratories that I manage. And they also perform trace back if there's an outbreak and we need to trace implicated product back. And then manage the logistics of recalls, which are really sometimes quite complex. And then there's the Office of Regulatory Science, where I'm based. And we provide laboratory support for surveillance sample testing, outbreak sample testing, results communication, and coordination with the FDA centers like CDN and CIFSAN and CORE. There are counterpart scientists, peers and colleagues in laboratories within the centers that we work with on very closely for not only outbreak investigations, but also technological advancements and method development and so on. So the nuts and bolts of an outbreak investigation. I'd like to emphasize that I can't say more about our state partners and how important they are in the foodborne illness outbreak investigations that we perform. They're usually, FDA gets involved when it's a multi-state outbreak investigation. We always end up collaborating and coordinating with our state partners and the CDC. But it's a coordinated effort that not only to address outbreaks as they occur, but to help mitigate the impact of these outbreaks and to prevent outbreaks from ever happening. There's epidemiologists that are helping with the investigation. There's inspections. There's sample trace back. There's implicated product distribution that needs to be traced. And then, as I mentioned, managing the product recalls. The goal is early identification of illness clusters and then rapid and effective regulatory response to remove contaminated food from distribution, and then outreach to inform consumers. And so although foodborne illness remains a significant overall burden to human illness, there is good news that technology and enhanced response capabilities have reduced the size of most of the outbreaks that we're working with now, as in fewer cases of human illness. And in general, the outbreaks are detected sooner than they were in years past, and that's largely due to some of the genomics and metagenomics technologies that Dr. Maiden mentioned previously. A little bit about FDA CORE. So it was established in 2011, and it brought together expertise in medicine, public health, epidemiology, and science to coordinate efforts to identify, stop, prevent foodborne illness outbreaks. And so when an outbreak is associated with an FDA-regulated product, there is a response team stood up in CORE that coordinate FDA's response efforts. And as I said, in any given day, there's usually multiple outbreaks that we're working on, and staff in my laboratory organization interact with staff from CORE on a daily basis, and it really is a productive collaboration. And to give you a sense of CORE's contributions since they were stood up in 2011, and these numbers grow almost a daily basis, but been working on up to 1,000 potential outbreaks, responded to over 200 outbreaks that potentially linked to FDA-regulated products. They identified contaminated food products in over 100 outbreaks and warned consumers to avoid implicated products through more than 400 public notifications. Over 251 food recalls are initiated and multiple targeted assignments, including for food facilities, farm investigations and inspections, record collection, and sample collections related to these outbreaks were initiated by CORE. But as I said, the C in CORE is coordinated, and so it's a coordinated and collaboration among multiple partners, including within FDA and external to FDA. So this is a list of some of the primary foodborne pathogens that we work with. There are certainly other organisms that we encounter, but these are the main ones. And to link this back to Dr. Neal's presentation on One Health, most foodborne pathogens are zoonotic pathogens, and so the interventions that we take largely take a One Health approach, and I'm certainly an advocate of One Health to addressing the burden of illness in both humans and animals. And so E. coli 157H7 is an important one that we work with. Most of the outbreaks of late have been linked to leafy green, primarily lettuce. Salmonella enterica, Listeria monocytogenes, and then the protozoan parasite Cyclospora cayemensis, and some viruses, hepatitis A and norovirus, which we've had a number of outbreaks linked to those in recent times. So some recent investigations, FDA has provided technical and epidemiological and inspectional laboratory support for several recent outbreaks of foodborne illness. I'll start with E. coli 157H7 and leafy greens. But I mentioned Cyclospora in fresh produce. That's primarily imported produce that we see that. Salmonella enterica in a variety of products, and we're working on an outbreak right now in a cashew brie product. And then hepatitis norovirus in fresh berries and produce. Again, those are usually in imported products. So on the E. coli 157H7 front, we've had ongoing investigations in the lettuce growing regions of the U.S. In 2020, there were 2 outbreaks that were multi-state of E. coli 157H7, and epidemiology and traceback determined that they were linked to leafy greens. And when we looked at the whole genome sequence analysis, the genetic fingerprinting of these isolates, we could see that they aligned with the cases that we were seeing in 2020, but they were also associated with prior outbreaks in 2018 and 2019. And so because of this reoccurring incidence of this particular strain of E. coli in leafy greens, primarily lettuce, the FDA has initiated several sampling assignments that were focused on the growing regions in Arizona and California, looking at both environmental samples and product samples for this pathogen and others, including Salmonella, and there's other hemorrhagic E. coli of interest as well. And in fact, there's an assignment going on right now, a surveillance assignment, which is looking hopefully to prevent occurrence of illness by detecting it before the product gets to market in the Sal Bowl, California, targeted towards leafy greens and lettuce. Another recent outbreak investigation late last year and into early this year involved Salmonella poxstem illnesses. There were 7 cases in 4 states. We were notified by CDC, and then we stood up CORE to help coordinate the investigation. The epidemiology implicated smoked fish. Pretty quickly, FDA was able to identify a firm in Brooklyn, New York that manufactured the product, and it was linked back to tuna that was imported to the U.S. from Indonesia. So FDA collected 15 product samples and 1 environmental sample and submitted them to one of the labs in my shop. And at the time, put the Indonesian firm on increased screening. And after testing the product, we found that 4 of the samples collected had Salmonella in them, and the positive samples and products were from the Indonesian supplier, and the isolates aligned with the outbreak strains that were being seen. And so the FDA has some tools called Import Alert and Detention Without Physical Examination that we use for imported food products, and they're excellent tools that allow us to prevent contaminated food products from entering U.S. commerce. And so this particular firm was put on an import alert to keep tuna from this supplier out of the U.S. until they could demonstrate that their product was salmonella-free. Another recent investigation was on Listeria monocytogenes in Spanish-style fresh and soft cheeses. Again, it was a multi-state outbreak investigation. CDC had the initial signals and stood up a CORE. The FDA did an investigation. Samples were collected and tested. We worked closely with the state lab that was initially involved in this. And Listeria monocytogenes was isolated from product. The product was recalled, and the case has since stopped, but whole genome sequence analysis was used to determine that the product found in the cheese samples aligned with what was being seen in the humans. And I'd like to emphasize the importance of Listeria monocytogenes, especially in ready-to-eat products. It's one of the least common foodborne pathogens, but it has a very high case fatality rate. So it's really a high priority organism for us to detect and then remove contaminated product. And so on the radar, you know, on any given day, we're looking at signals to see what's going on and if outbreaks are occurring, and FDA and CDC are continuously tracking clusters of foodborne illness, and then we apply epidemiological tools to detect outbreaks. So currently tracking a few potential outbreaks without a known food source yet. I mentioned there's one active outbreak in cashew brie where product has been recalled. It was linked to salmonella. And so, you know, collectively, FDA, state and local food protection agencies perform risk-based inspections and food sample collections. And a lot of these efforts are preventing outbreaks that we never even know about. And then we have the tools and resources in place to respond to outbreaks as they occur. And with that, that is the end of my presentation.
Thank you, Dr. Rice. Dr. Edwards, can you please turn your camera on? And you may begin your presentation once your slides load. Thank you.
There we go. All right. Good afternoon, everyone. And thank you to the organizers for this invitation to be part of the session on innovation in food and cosmetic safety. So as you will soon see, what I'm going to talk about today really builds upon what other presenters have already discussed. When we talk about One Health, we know that livestock, poultry, and pets, all ill and mental health of people. Working together on One Health does help across the human, animal, and environment to maintain the healthy ecosystem that involves us all. Seeing this beginning slide, how many with pets at home have wondered the question posed here? It's really astounding the number of different ingredients that appear in pet foods today and or things that pets may ask us for around the house. In the division of animal feeds, we see pre-market as well as post-market reviews for ingredients that go into animal diets, not only of pets, but those of livestock, poultry, and aquaculture species besides. The submissions become more and more complex every year, and yet we have to keep up with the innovations in order to ensure the safety of human and animal health. Although a few years old now, the data on this slide indicate the impact of those innovative ingredients and the mixing of animal diets in the U.S. The dots on the map indicate animal food manufacturing facilities. From the east to the west across the country, animals are consuming almost $300 billion worth of animal food each year. As a reminder, different from humans, the domestic animal species tend to consume very similar diets for much of their lives. This is one of the big considerations in assessing the safety of animal food ingredients. It is also important to consider that a food an animal consumes today may become the meat, milk, or eggs are on our own plate tomorrow. Animal food is manufactured in every state in the United States and includes those foods that are made for all different types of species. It continues to grow and makes a big impact both on our mental as well as our physical health. Now here's a quick quiz. You thought you probably wouldn't get a quiz today, but some of the animal food ingredients that we see may sound foreign from what we see on our human food labels. In reality, though, some of the terms have been around for quite some time. So here's the quiz to remember and ponder as I go through this talk. Does anyone know the definition of bagasse, corn gluten meal, or de-oiled corn distillers dried grains with solubles? All of these are animal food ingredients used on a daily basis. And so stay tuned for answers to this quiz later in the talk. You don't have to come up with the answers all on your own. I think many of you are familiar with the Code of Federal Regulations, as well as the Food, Drug, and Cosmetic Act. Obviously, in the Act, food is defined as articles used for food or drink for man or other animals. Any substance that's intentionally added to an animal food must either be an approved food additive listed in 21 CFR 573, 579, or a substance that's generally recognized as safe GRAS for an intended use. In the animal food space, in coordination with state feed control officials, which we've heard of the coordination in other talks today, CVM also recognizes ingredients in the AAFCO official publication as being acceptable for use in animal foods. Now let's talk a little bit about some of the innovative ingredients and some of those ingredients that have been utilized in animal foods for a long time. Byproducts of the production of human food have been used for centuries to feed animals. Examples appear on the slide, things such as the wheat middlings that come from processed wheat for flour, those peels, rinds, and pulp that might be left over after the processing of fruit and vegetables. And now today, recycled foods or foods that maybe past their sell-by date but still be safe, things such as chips, cookies, candies, or bakery products. And they end up not going to the human food supply due to quality characteristics, but yet they may still be safe. These human food byproducts can be either in a typical commerce stream or can be a one-off stream where you don't see those all the time. Today's animal will include these byproducts, but they may be processed ways or have quality defects that do not impact safety, as I mentioned. There are several initiatives underway, both in the public as well as the private realm, to lessen the food that is thrown away in a landfill. And one of the options that is mentioned often is that food being utilized in new ways as animal food. Obviously, these different products still need a safety determination to ensure that the animal food is safe and any resultant human food from those animals. We have seen many new products and different fractions of products over the last few years. Another place that we have been looking at the promotion of innovation and emerging technologies and product safety are things that you see on this slide. Most simple animal food ingredients have already been approved in one way. I think that those would be called the low-hanging fruit. The submissions that come in today for us to review are complex and have things such as genetically engineered organisms producing specific thermostable enzymes come in today, not such low-hanging fruit anymore. As livestock producers move away from antimicrobial drugs administered in feed, they'll be looking to provide optimal animal nutrition and support gut health with things such as enzymes, probiotics, and prebiotics. This is a prevention approach versus a treatment approach, and the healthy animals through diet rather than through medication. So we've been successful in moving towards this judicious use of antimicrobial drugs. We've also created a pressure point where we're going to see an increase in novel and complex animal food ingredients. Adding to the pressures that on animal food ingredients are things such as consumer preferences. These consumer preferences may indicate where they're asking for certain ingredients to be included in animal diets, or on the flip side of the coin, for the absence of certain ingredients within animal diets. CVM will see more food ingredients coming in for pre-market review with these new additional ingredients. CVM reviewers do receive more data to dig through to adequately assess these new and complex ingredients. For both the animals eating the food ingredient and the humans eating the edible tissues of those food-producing ingredients or food-producing animals, it's important to look at the safety. The couple pictures that are shown here on the slide are two new ingredients that have come through for animal food in the recent years. The top picture is that of black soldier fly larvae. This is a source of protein and oil for things such as poultry, salmon, and maybe soon other animals. These insects are raised on food scraps, which would otherwise have gone to waste. Instead, the insects eat the food scraps and are turned into high-quality food for other animals. CVM looked at the fatty acid profile to see if it matched those needed by chickens and fish, and thus determine safety and usefulness of the ingredient. The lower picture on the slide would be that representing marine microalgae. This would be single-cell algae farmed to produce DHA for dogs, thus saving pet owners from having to get it from fish taken from the ocean. CVM also looked at safety for those dogs and whether the DHA would be useful in the dog's diet. A couple examples of ingredients that have come through CVM in the past several years. While looking at these different ingredients, obviously safety is an important part of the review. The safety of an ingredient does differ amongst species, though. Our safety reviewers in my division really do like to remind me that a dog is not a cat, is not a rat. When we consider safety, we do so across many species, including the safety of any human food that the animal may be producing. Earlier, I talked about human food byproducts being a part of the animal diet. Additional considerations for safety need to be taken into account due to these species differences. A couple examples here of some human foods and minerals that have been found harmful in certain animal diets. You might not expect this across the different species, but with those differences across the different species, you see things such as xylitol, which appears in many human foods but is harmful to dogs. Things such as sulfur amounts to harmful to cattle, and copper can be harmful to sheep. Safety is key. What an animal eats today may be the food you eat tomorrow. As we look into the crystal ball in terms of what things might be next in line for animal food ingredients, that innovation really is taking many different shapes. Some of those trends are easier to predict for others, but here are a few that I'll list as we're looking to the future. You may have heard of cell-based animal foods mentioned in an earlier talk today, things such as hamburgers grown in cell culture, but the same is also happening in pet food. One innovation that we have seen is to grow mouse cells up to be part of cat foods. It's also occurred where growing other traditional livestock animals in a cell-based culture for other types of pet foods, for dog foods and for cat foods. So one of the interesting new innovations. We've all heard a lot about hemp and hemp seed ingredients. These two are occurring within the animal food space, both the hemp seed oil as well as what's left over after pulling off the oil, the hemp meal, are things that we'll see. We've heard of shareables. Now, shareables is not necessarily a new ingredient per se, but one in which an ingredient or a food may be something that both the human and its pet could eat, where you could share that with your pet. We're not so sure about the safety of these and haven't had to look at those yet, but that is something that may be coming, where we're evaluating not only safety to the animal, but also safety to the human, because they're both eating it together. We have seen innovations where pet foods would be intended to be reheated at serving. So you not only would heat the food for yourself at dinner time, but you would heat the pet food, hopefully not on the same plates, and hopefully being safe between those. But that is something that has come, and it's part of the safety consideration if you're looking to make sure that you keep safe for both the pet as well as the human who's handling those foods. And I think I talked a little bit about it, but taking today's food waste and feeding it to livestock is something that's been done for a while. But new components and new human food leftovers are things that are making it into the pet food and livestock food supply. Some examples of this is where human food leftovers would be mixed together and enzymatically digested to create part of an animal's diet. This would help with furthering the reduction of food waste here, and it is something that lots of firms have been talking about, and we've been thinking about how to do the safety recognition of that. So now, does anybody remember the quiz from the beginning? If you have looked up the answers on Google in the interim, or if you know the answers and we're thinking about them, here's your quiz answers for today. So bagasse. Bagasse is that portion of the stalk of sugarcane after removal of leaves and tops that remains after extraction of the juice. The gas can be used as a source that has a lot of fiber in it and is utilized in different animal diets. Did anybody know what corn gluten meal was? Well, it's the dried residue from corn after the removal of the part of the starch in the germ. Separation of the bran by the process employed in the wet milling manufacture of cornstarch or syrup, or by enzymatic treatment of the endosperm. And it doesn't have any wheat gluten in it. So when you see corn gluten meal in your animal diet, it's not quite the same gluten as you would see in wheat gluten. And the last one, the de-oiled corn distillers dried grains with solubles. Now that's quite a mouthful, but it's a portion of the corn that is resulting from the solvent extraction of oil from corn distillers dried grains with solubles, which come from ethanol production to result in a crude fat content of less than 3% on an as-fed basis. And it's a source of protein that we see in many livestock diets. So today, hopefully you have found out a little bit about some of the animal food ingredients, learned a little bit about some of the innovations that are coming down the line, and remember that One Health, talking about the health of animals and humans and the environment, are all tied together through the safety that is looked at. I appreciate the opportunity to be a part of this panel, and for further information you can visit the website on this slide. So thank you very much.
Thank you, Dr. Edwards. Dr. Camacho, can you please turn your camera on? And you may begin your presentation. Thank you.
Thank you. So good afternoon. Thank you, Chad, and good afternoon, everyone. I'd like to thank the organizers for the opportunity to showcase some of the activities that we are conducting at the NCTR to contribute data to evaluate cosmetic safety. So what I'd like to do first is to give you a very brief overview of our centers, the National Center for Toxicological Research, and then showcase a couple of examples of studies that we've conducted to provide safety data on different cosmetic products and ingredients, in this case tattoo and permanent makeup ink, as well as aloe vera leaf extract. And then a new study that we started a couple of months ago that is evaluating a novel tool that may be of use for in vitro permeation testing, for instance, of cosmetic ingredients. So NCTR, or the National Center for Toxicological Research, was funded 50 years ago, almost to the date, and it is a non-regulatory research center of the FDA. So unlike the other centers, we are located in Arkansas, south of Little Rock, and we share the campus with the Office of Regulatory Affairs, Arkansas Laboratories. So the campus is quite wide. It includes about 30 buildings, and we have a staff of 550 plus members. And these include full-time researchers, staff fellows, and postdoctoral fellows, as well as a number of on-site contractors, including animal care and pathology contractors. So the NCTR mission is twofold. So we conduct scientific research to generate data for decision-making by the FDA product centers, as well as we develop and support innovative tools and approaches that the FDA can use to protect and promote individual and public health. And so in this sense, NCTR supports all offices and the product centers at the FDA to support their regulatory mission and decision-making. So we have quite a broad scientific expertise, and I hope you had the opportunity to listen to some of the talks of my colleagues over the yesterday and today on AI, bioimaging, microbiome studies. Here I'm just listing some of the studies more focused on that we've conducted over the years, more focused on the evaluation of cosmetic safety. And these include rodent toxicological studies, including chronic dermal studies, both in vivo and in vitro skin permeation studies, pharmacokinetic studies, biodistribution studies, as well as a wide variety of genotoxicity and mutagenicity studies. And we also conduct and have expertise on the chemical and microbiological analysis of cosmetic products and ingredients, for instance, to characterize their heavy metal loads and potentially identify pathogenic agents. And so what I'll like to do first is, and here I'm just showing some of the chemicals that we've evaluated over the years on ingredients that are widely used in skincare products, for instance, alpha and beta hydroxy acids, aloe vera, retinyl palmitate, more recently CBD or cannabidiol, as well as permanent makeup pigments and tattoo inks. And many of these studies, in particular the genotoxicity and the bioassays, have been conducted often under the sponsorship of the National Toxicology Program through an interagency agreement between NCTR and NIHS DNCT. So the first study that I'd like to share with you today is an ongoing microbiological survey of tattoo inks and permanent makeup inks. And this study is being led by my colleague Seong-Jae Kim at the Division of Microbiology with the support of the CIFSAN's Office of Cosmetic and Colors. And this study was really initiated because about 10 years ago, there were several outbreaks of tattoo-associated skin infections with pathogenic bacteria. And so this really raised awareness and recognition that tattoo inks can be a potential source of bacterial infection. And so what we've been conducting at NCTR for a few years now is a survey of an open tattoo and permanent makeup inks that are sold in the US and conduct an evaluation of the microbial contamination, including of pathogenic bacteria. And there's been a couple of reports published already, so I would encourage you to look for them if you are interested to learn more. Just to give you an idea of the findings, here I'm showing the geographical distribution of the manufacturers from which products were evaluated this far. And this includes a wide range of manufacturers both in the US as well as imported products. And you can see that there was indeed widespread microbial contamination of the product. So in red, I'm showing the number of the products that were found to be microbiologically contaminated versus the total number of products. And you can see that the red really stretches around the map. When looking for the identity of these bacteria and as well as fungi were identified, most of them fall on the genera, species from the Bacillus genera, and about half were potentially clinically relevant isolates. However, no mycobacteria were detected on the almost 200 products evaluated this far, but there were some recalls initiated by CIFSAN based on these data. So switching gears, I'd like to share also another study which we conducted to assess the photocarcinogenesis of aloe vera leaf extract in hairless mice. And this study was conducted and led by my colleague Dr. Mary Boudreau at the Division of Biochemical Toxicology, and it had the sponsorship of the National Toxicology Program. And all the study report was published in the NTP Technical Report Series, and it's Product Report 553, in case you would like to learn more about this study. So as you are probably most aware, aloe vera leaf extracts are widely used as a cosmetic ingredient in a wide range of cosmetics and skincare products, ranging from lotion, shampoos, and wipes. However, there was uncertainty on whether aloe vera in skincare products could enhance the induction of skin cancer by UV radiation. And so we conducted a one-year photocarcinogenesis study to evaluate whether the topical application of various aloe leaf extracts could alter the effects of simulated solar light on the skin of hairless mice. And we tested different extracts, including the whole leaf extract, the decolorized whole leaf extract, as well as aloe gel. In the next couple of slides, I'll show you some graphs with the study findings. I'm going to show the female data on the left and the the male data on the right. And here it's listed the neoplastic skin lesions that were observed in the rats, both individually and all combined. And in this slide, I'm showing the controls of the study. So as expected, the simulated solar light induced an increase of both the incidence and multiplicity of squamous cell skin neoplasms in the exposed rats. And this effect was dose dependent. So the longer the exposure to the light, the higher the incidence of the skin neoplasms. Also as hoped, the control vehicle cell had no effect. So here in black, I'm showing the control cream plus the simulated solar light, or just the solar light, simulated solar light in white, and you see there's really no difference between the two groups. When the cream was formulated with aloe vera leaf extract, we observed weak enhancing of the effect of the photocarcinogenetic activity of the simulated solar light. And so again, just to illustrate the data here, in this graph I'm showing the multiplicity of squamous cell skin neoplasms in the aloe gel groups, and you can see in the females, compared to the simulated solar light only group, we did have an slight increase of the neoplastic skin lesions. And this was observed in females exposed to aloe gel group, but not in the males in this case. And so the conclusion is that aloe leaf extract, when applied topically, can have a weak enhancing effect of the simulated solar light neoplastic activity in the skin. The last study that I would like to share today with you is a study that is being conducted with the support of the FDA chief scientist and in collaboration with the colleagues from the Center for Food Safety and Applied Nutrition, the Center for Drugs, and Center for Veterinary Medicine at the FDA, as well as colleagues from the NIH, NCATS, and the NISTM. And what we are evaluating here is the potential of 3D bioprinted human skin to be used in vitro permeation testing. So as we know, it is important to understand whether a compound that is applied to the skin, such as a cosmetic, remains on the skin, or by the contrary is absorbed through the skin to reach the bloodstream and potentially reach distal target organs. And so the assessment of rate and the amount of dermal penetration of cosmetic ingredients is a key consideration in their safety profile. In terms, both in vivo and in vitro testing can be performed, and in terms of the in vitro absorption studies, excised human skin is the current gold standard model. However, there's ongoing research efforts to develop and validate non-animal but human-relevant alternative models of native human skin. And so in this project, what we are evaluating is the potential of a 3D bioprinted human skin model that has been developed at the NIH NCATS in the laboratory of Dr. Mark Ferrer for in vitro permeation testing. And so NCATS is manufacturing the bioprinted human skin, shipping to NCTR, where we are conducting further characterization of the model, as well as in vitro permeation testing. And here I'm showing you can see the front cells, which are the setup we are using for the in vitro permeation testing, with the inserts with the bioprinted skin mounted. And so this is a new project, and hopefully in the upcoming months we'll have exciting data to share with everyone. And so this is where I will stop. I hope I was able to illustrate some of the efforts that we are conducting at the NCTR to develop and evaluate novel tools that can be used for cosmetic safety evaluations, as well as some studies that we have conducted to address regulatory data gaps. And thank you for your attention, and I'll be happy to take any questions. Thank you.
Thank you, Dr. Camacho. Excellent presentations from all of our speakers. I would now like to invite everyone to turn on their cameras, and we'll now turn it over to Dr. Jeff Ward to begin the panel discussion. Thank you.
Okay, thank you, everybody. Just waiting for Dr. Rice to join. There we go. Okay. I just told Chad that I was happy that we had more time than was allotted for this discussion. I think this could be the most exciting part. Thank you, everybody, for your very good, excellent talks. I think the people from the FDA always sort of relish these opportunities to reach out and talk to the general public and whoever else. And I know that we always relish reaching out and interacting with people like Dr. Neal to get their perspective on what we're doing as far as the science side of things go. So we have come up with a number of questions, which for full disclosure, we did share with our panel, just so there weren't any communication issues, so everybody knew what we were going to be discussing. So we tried to have some general questions that everybody could get involved with, although a couple of them are a little bit more focused than others. So to kick things off for the first question, and this is really sort of a One Health-oriented question for all the speakers. I think everybody could have input on this. We have all seen the impacts of diseases that affect many species, humans and animals. Dr. Neal and others have talked about COVID-19 and the issues involved there, but there are things like Ebola, which we know has an animal reservoir, obviously things like BSE, mad cow disease, and even diseases like African swine fever virus, which doesn't affect humans directly, but it does get into the wild populations. It has economic impacts, and it also has pretty big nutritional impacts because a lot of parts of the world depend on pork for their high quality protein requirements. So considering all those different zoonotic or other diseases that affect humans in one way or another, what else do you think that we can do to increase our ability to detect these pathogens or to prevent their spread in FDA-regulated products? And I'll let anybody who wants to jump in on that.
So maybe I'll just start us off a little bit. So what first comes to mind is, and I think many of us are having these conversations about the silver linings from the pandemic, right? What have we learned that we can take away, that we can really continue to invest in? And to me, one of those is wastewater-based epidemiological approaches to surveillance. And the FDA is very much involved in this and sequencing wastewater and looking at COVID analysis that way. The Centers for Disease Control and Prevention have launched a national wastewater surveillance system. And I think capitalizing on what academics and private-public partnerships are doing around the world to look for SARS-CoV-2 and using really increased diagnostics like digital droplet PCR, which allows us to have better precise detection, we can use that to monitor for almost all of the agents that you just discussed, the viruses, bacteria, certainly, probably a little bit difficult with some of the prions. We might have to figure out what we look for there, in particular certain proteins. But I really think that this infrastructure that's been developed literally around the world, and it's so exciting to see it start and populate and scientists really get involved. And what's really neat is even in the state of Delaware, we're using it with students and in middle schools and high schools, and I think we've got some people listening to us from those schools now. And these students are getting involved and they're learning about detection of these agents, from their own wastewater and around the surface water and everything else. And so I'd love to see us be able to continue to use those type of surveillance mechanisms to really continue to look for diseases before we see them in our populations.
And I guess I can go next from the FDA perspective. And I agree with what Dr. Keneal highlighted, and a lot of it's that public health infrastructure. And we have to invest in the public health infrastructure. We never know what's going to be right around the corner. But having labs that are ready to go, that are trained, that have sequencers available, that understand the bioinformatic platforms. One of the advantages of our Genome Tracker network is that we have these labs around the states and also in other countries. They know how to share the data. They know how to upload the data into NCBI so people can access the data around the globe in real time. And it's taking advantage of those kinds of networks and experiences that are so critically important. I do agree about the wastewater issue as well. We have been using wastewater surveillance as part of our foodborne pathogen work. So we've tested wastewater for things like norovirus and Hep A and cyclospora as well. And so that is work that we have developed, including developing analytical methodologies to pull out things like cyclospora out of wastewater. And now we are utilizing those same skills from genomic sequencing, from wastewater surveillance, and we are doing some work with COVID variant detection as well. So it's taking these investments we've made in the public health infrastructure, making sure those labs are current and functioning and know how to do this work, and being able to deploy them as we end up with any future public health challenges like we've had with SARS-CoV-2.
Yeah, and I'd like to jump in. This is Dan Reitz. I really appreciate the comments by Drs. Neal and Main. And I agree, a robust global public health laboratory network is going to be key with the One Health approach. I'd also like to highlight technology in our laboratories as well. And so 10 years ago, we were all really excited about whole genome sequencing. It's pretty routine in our labs now and has been just a really fantastic tool. The next thing to come is going to be metagenomics, and what I think I'm looking forward to is the day that we don't have to culture confirm and isolate to take regulatory action, that we have actionable data based on the sequence data. And metagenomics is promising to move us in that direction.
If I can just jump in, I just wanted to raise awareness that we are actually at NCTR conducting a study on looking at the wastewater SARS-CoV-2 detection, and we've collected samples for over 1 year now. So we started really early on since April last year, and we've been collecting samples every 2 weeks. And actually, we have a poster. So if you are interested to learn more about it, I would really encourage you to look for the poster. And it has been a very interesting study. We really followed the time profile of the cases, and we are working with the Arkansas Department of Health to try to relate to reported clinical cases. And yeah, it just shows how powerful that technique of community level surveillance really can be, not just for COVID-19, but to other pathogens in the community.
And one of the things I'd like to mention too is the things that we've learned over this past year about biosecurity on the human scale, whether it's social distancing, vaccination, or the wearing of protective coverings. Those are things that are good to be a part of whatever biosecurity it is, whatever disease it is. Well, we see in modeling for things like African swine fever, where the spread of African swine fever may be within feed, but it may be attributed to feed, but it may actually be on feed trucks, or it may be through feed totes, or things that are associated with the feed. And I think it's likely that something like that probably comes in on a person, and then maybe spread might be through machines and that kind of stuff. But biosecurity is something that is not only applicable to the animal space, but it's also applicable in the human space that we've all experienced over this last year.
Great, and as sort of a follow-up to that question, and everybody on the panel knows that the FDA isn't the only player here. There's the USDA, there's the EPA, there's even the Department of the Interior and the Fish and Wildlife Service, and there are a lot of different players involved here. Are there any particular issues involved on that side as far as interaction, communication between parts of the government that could be improved, where you see that there are holes that need to be filled? And maybe the answer is no. I don't know. But it's it's really more of a global sort of a One Health approach to it?
I think with communication, the answer is always yes, there's room for improvement. I don't have any personal examples, but I know that, you know, we struggle with really timely and effective communication, and we have for years, and we keep trying to improve that, but there's always room for improvement for collaboration and bringing down the silos and communicating information.
And I would echo that, Jeff. I think it also depends on like what our regulatory authorities are. And so when it comes to things like contaminated water, if that's a risk that could then contaminate food, EPA regulates the water, not FDA, so we would need to work with them in that particular space. So the different government agencies have different authorities. But in a One Health perspective, what that means is we all have to work together. And that includes USDA, that includes EPA, that includes us at FDA, but also CDC and others. So I think that's another thing we've learned through the pandemic is the amount of engagement we have had with other federal agencies. In the setting of SARS-CoV-2, that also included folks like the Department of Labor and OSHA, and we're working to protect farm workers and others who were at risk, both for themselves, their families, and their communities, but also that would increase the risk of food supply chain disruption. So the amount of engagement we had to mount during SARS-CoV-2 was certainly illustrative of the importance of working together in a One Health way when we have a public health emergency.
And I think Dr. Main's comments are spot on. We can always have conversations and tabletop exercises and those things, which is very valuable in preparation for emerging diseases and things like that. Obviously, in this last year, we've had one of the world's biggest tabletop exercises in terms of how we've dealt with this. And those lessons that we've learned and the times that we've had to work with these other agencies can be lessons that we can take forward.
Okay, great. Moving on to the next question. And I'm going to put a caveat on this one just because I'm a veterinarian. My wife is a small animal veterinarian. So I'm going to talk about something that's more cosmetic related. But just so people know, people use Avon products on their pets for insect repellent purposes. People put nail polish on their pets. People tattoo their animals for identification purposes. So there is probably more crossover here than a lot of people are aware of. But the question is this. The FDA is putting a lot of effort, a lot of resources into developing new methods or alternative methods, as we call them, to help reduce the amount of animal testing as it applies to food additives, packaging or food contact materials, cosmetic ingredients. So this is really sort of focused on Dr. Main and Dr. Camacho. How does this approach as far as reducing animal testing or increasing alternative methods, how does it fit in with FDA's predictive toxicology roadmap, and especially with the new technologies that NCTR is working on for safety evaluation for different products?
Maybe I can start and then Dr. Camacho can jump in. Yeah, the first thing I'll say, it's not unique to cosmetics. At the FDA, we really want to reduce the reliance on animal toxicity testing across the board, as you highlighted, for food ingredients, for anything that we regulate it. And the other point I'll make is that we want to get it right. We want to know what's the most predictive of human toxicity. And you can test compounds in 2 different species of animals and get 2 different answers. So it's not that animals always give us the best prediction. So I think what we're trying to do is really look at the totality of the science and use all of the tools that we have available. That may be in silico methods. What do we know about chemicals? What do we know about their structure? What do we know about what metabolic pathways they impact? What does that tell us about potential routes of toxicity? What in vitro assays do we have where we could screen large number of compounds at the same time? Are there non-mammalian systems that we can study that can really tell us something about toxicity? And then when we look at all of that evidence together, how do we get the best prediction about human toxicity risk? So some of the work we're doing in CIPSAN includes things like we're studying C. elegans. It's a nematode. It's a little teeny tiny worm. We're looking at that to see how that can model effects on neurodevelopment, neurobehavior. And these are, again, non-mammalian systems that could help advance much of the work, where the mammalian systems have been especially challenging to predict human toxicity in this area. We're also, like many other laboratories, working on microphysiological systems, organs on chips. And that is, can we do these types of methodologies looking at specific organs, whether it be lung or liver or gut, to really be very predictive of toxicity we would see in the human setting. So I think the whole thing is building the foundation of science so that we can do the best job possible predicting human toxicity, and it doesn't have to all revolve around animal models.
And I'd like to echo Dr. Main's words. So I agree, it's not just cosmetics, it really spreads across all the products that are regulated by the FDA, and really the effort to develop, validate tools that are alternative to the use of animals. I know it's a very important and critical effort by the agency, but again, we have to do it right and to build enough robust data that we can do the jump for those alternative methods. And so NCTR really has a lot of active research. As I mentioned, part of our mission really is to develop and validate innovative tools that can be used to support the FDA regulatory mission. And in all these projects, we work very closely with the colleagues from the product centers exactly to make sure that our work really goes towards their needs. And so in my talk, I mentioned one example of a study that we are conducting to try to assess whether bioprinted human skin can be a useful tool to replace the use of animal skin as well as human native skin for in vitro permeation testing. But we have, of course, other studies, including several looking at microphysiological systems such as placental, testes, liver, as well as also computational tools that can also be able to provide data without the resource to animals. And so, between all these activities and the close interactions with the centers, we hope we can contribute to the implementation of the FDA predictive toxicology roadmap.
Okay, great. Moving on to the next one, which is a wide open sort of area to talk about. And I think many of you addressed this in your individual presentations. But, Dr. Main, Dr. Rice, Dr. Keneal, and I think Dr. Edwards, even you and CVM, who are using these approaches as well. Can you talk a bit more about the impact of the really revolutionary combination of whole genome sequencing, genomics, metagenomics, bioinformatics, as it relates to foodborne outbreaks and also in preventing and mitigating human or animal illnesses or deaths? And anyone who wants to start.
Sure, this is Dan Rice. Yeah, that's a great question. And so I remember I was moderating a session at the Association of Public Health Laboratories annual meeting about 10 years ago. We had a panel of people. Eric Brown from Genome Tracker was there. Some of the PulseNet people were there. And that was back in the day of PFTE, pulsed field electrophoresis, and we were talking about what is whole genome sequencing going to bring, what's it going to do for us? And so now, 10 or so years later, it's a routine part of what we do. And there's really interesting data from CDC, the PulseNet group that I've seen that demonstrate the value of these technologies and identifying outbreaks quicker, reducing the size of the outbreak. So right now we might have more outbreaks. It might seem like we have more outbreaks, but we're detecting more of them than we used to. And the overall number of cases is going down. And that was just with whole genome sequencing. And so, who knows what the future is going to bring with metagenomics and the technology that's coming our way. It's just amazing how we just expand our capabilities exponentially. So I'm really excited to be in the area I'm in now and not close to retirement, so I can, really enjoy what the future is going to bring, and I'll just leave it at that.
And I can give some perspective as well. I think, you know, when people think about outbreaks, we often used to think of it, you always start with the epidemiology, you figure out who's got the disease, use whole genome sequencing to link those cases together, even if they're geographically dispersed. Then you ask people what they ate, then you try to figure it out, and then you go test product following the epidemiology. But I think what we see now with whole genome sequencing is sometimes we'll find cases and we'll do the whole genome sequencing and we'll find that we have a cluster of illnesses and we'll look in the database and we'll have a clue to a food source before we've even done the, you know, the epidemiology piece of it based upon the sequence of the pathogen, because we've seen that pathogen before. We've seen it in a particular region. We've seen it in particular products. That doesn't give us evidence of causation, but it certainly gives us a head start in terms of trying to figure out what's going on in outbreaks. Another example I think is that we have learned that I think is really important for prevention is we have seen facilities that have produced contaminated food, and then we start sampling in the facility and we do environmental testing in the facilities and we find out they've got a pathogen in there and that's led to contamination. And then we go back, 6 months later to see how, how the facility's done following a contamination event. And we find the same pathogen again, and it's the same genomic strain, and then we know they've got a resident pathogen in there. That is a very, very serious food safety risk, and we want to address it. And our ability to find that and to know that they've had an ongoing problem with the same pathogen is really important in how we address prevention of future outbreaks. This is not a sporadic introduction of a pathogen. This is the same pathogen coming back over and over again. So we use that in the prevention piece of that. And in outbreaks, it's not just about getting contaminated product off the market. That's obviously, job one, but it's making sure it doesn't happen again. And whole genome sequencing is a tool that really can help us in that regard as well.
I think, too, one of the important pieces that we can look at with the whole genome sequencing is that preventive measure, where we maybe be finding that in a finished diet or in a pet food or something like that. But if you find that same pathogen and you've characterized it through some of these different techniques, you can trace that back into the ingredient and really find those other lots that might be a big part of, And so we've seen probably more expanded recalls in the last few years because of this. Because we are able to trace that back through. And it's not just, oh, it must just be this lot that's contaminated. You can also do a lot of that testing and figure out what things maybe need to be recalled before they even get out there, which I think has been, although it's hard to track some of that sometimes, it's been a real plus on our prevention of causing those issues and those pathogens from getting out into the general population.
So, Dr. Kniel, if I can put you on the spot, how is sort of the One Health area utilizing this part of science, the whole genome, using genomic methods for tracking things, or associating different things across the spectrum?
Yes, it's really critical in the development of the ease of implementation of not only the technology, but also the data has really advanced over the past couple of years. And it's more available now across, again, private and public partnerships, not just federal labs, right? And I think that's really an important part of it. I certainly echo what Dr. Main said, which is so important is that prevention piece. And I've worked with producers who have had contamination events. We are able to sequence those and show them that it's a repeated offender, so to speak, and that really helps with the educational component of that. I think to get to your question, Dr. Ward, it really, when we think about some of the research aspects that are happening, we can use whole genome sequencing and even some metagenomics to understand why certain organisms are in an environment. Is it just a niche where they're, very comfortable, they have the right nutrients? Maybe it's a sanitation issue if it's outside of an environment like a surface water, or is there a reservoir for that organism? Certainly we've had this discussion and debate even with salmonella for many years. where it survives in multiple environments and low moisture foods as well. Foods that we're not really certain about and why do we have heat resistant microorganisms sometimes, but looking at the genomic analysis can really be very advantageous to help us understand why some of these organisms are there and relate them back to other situations. And again, as was mentioned, these databases that are so rich with these genomic sequences can be very helpful in helping us understand, where one organism was seen previously and now it's there. And I've been lucky enough, or I guess I feel fortunate to be able to work with the FDA scientists. Maybe the growers aren't so fortunate, but when we find an environment or that has salmonella, we find that organism, it's in Genome Tracker, and I get a call and saying, we found something that was just like that organism you found before. Can you tell us more about that environment? What animals were in that area? Where is there high poultry production there? Or were there reptiles? And so I think those discussions are really critical as we move forward to better understand, again, that One Health approach, what's going on in the environment. I think it's very exciting to see these tools really being used in so many different approaches.
Great. Very good. So the last question that we've come up with, and I might have to come up with more while you guys are talking since we're ahead of schedule. And this is I think really going to be a Center for Food Safety and CVM question, but I'm sure Dr. Rice, you can chime in, and Dr. Neal as well. And it has to do with the role of sort of intersecting or overlapping supply chains for human and animal foods. You've got byproducts or different components that go into both streams or that overlap. I think Dr. Edwards addressed, there are certain byproducts that come from the human food side that go into animal foods in some cases. So what role does sort of that intersection or interaction play in, for example, salmonella infection? And if something does come up, how are those issues detected and tracked, and can it be traced back to the other side? Meaning if something shows up in animals, can it be tracked back to possible input into the human food supply side of things? So I'll leave that open to anybody who wants to respond.
I'm happy to start, Jeff, on this particular one, and I can give you one example where this kind of happened, and that was with the Peanut Corporation of America peanut butter outbreaks in 2008 and 2009. And so what we had was contaminated peanut butter that led to a lot of human food recalls, and then that subsequently led to recalls of pet food that was found to contain the peanut butter paste that was used in both human and animal food products. So the issue was first, found out in the human setting, but it uncovered a potential hazard for pets. And that is when we find a contaminated product like this, part of what we try to do is figure out where the contamination occurred and then do like a trace forward analysis to see where that supply might have gone elsewhere to make sure that other relevant products are being recalled. And in this case, the human food setting led to animal, recall of pet food as well. And I'll say from an FDA perspective, CIPSAN and CDM work very closely together on these types of scenarios. Anytime there's a potential intersection between human and animal food, if we see an ingredient in human food that may have caused illnesses, if the supply chain leads to an animal food, CVM will be quickly looped into any of those kinds of discussions. So that's a real-world example where we've seen that kind of thing happening. So perhaps CVM wants to comment as well.
Absolutely, and I think that one of the things that we all know and something to remember is how closely tied together we are as a human and animal population. Whether it's our pets that live in our homes and the comfort that we give them and they give us, or whether it's the animals that are producing our food, we end up with a lot of the things on our plates having components that then also end up on the animal plates. So Dr. Main's example from the from the salmonella outbreak in the peanut products is a great example where we really needed to ensure that food safety is a big part of that One Health concept, that safety of human as well as animal health. And controlling those hazards in one part of these ingredients probably leads to safety of both humans as well as animals, because a lot of these components, I can think of many components of human and animal food that get split from the same essential origin. And so we see those things that way. The other component I want to mention here is that as we have things like industrial production, whether it's of ethanol and there are corn products that are coming from that, or other types of things, we need to keep those folks thinking about food safety as well. It's not only the production of whatever their primary product is, but it's the production of those co-products that is so important to both human as well as animal safety. We are lucky within FDA to have our folks in the Office of Regulatory Affairs aligned on both the human and animal health, animal food front. So those folks can very quickly be able to process through any problems that are happening on one side and think about how that occurs on the other side of that coin. And so I think that's one of the vital things. Obviously, the work together between our response teams in CISTAN as well as CVM is critical to this. And I think we, going forward, will probably have more and more overlaps as we are able to utilize some of the tools we talked about in the last question, whether it's the whole genome sequencing or others, to be able to prevent some of these outbreaks and hopefully find that in both the human and the animal front.
And the last point I'd like to add is, humans don't have to eat the food in order to get sick from the pet food. And Dave can speak about this issue with, contaminated animal foods, raw pet foods, things like that, and human illnesses that have ensued following that type of exposure as well. And you heard Dr. Kniel talking about the backyard chickens and things like that. So animals can contaminate, and including animal food, can make people sick even if they're not ingesting it directly. So another potential link between animals, their food, and human illness.
Great. Okay, so now we're going to go off script a little since we still have 8 minutes to fill. So these will be questions that we haven't shared with the panel, but I think that's fine. I think just about anybody can chime in on this. So on the human side of things, the FDA has FERN, the Food Emergency Response Network, for handling human food-related outbreaks. We have a laboratory network set up and a very elaborate response network. On the veterinary side of things, there's VetLearn, which is the veterinary lab network, which handles more animal-related issues. And I'm going to put Dr. Kniel on the spot here, and I know she's the one person outside of the agency. She's with University of Delaware, but I don't know that she has anything to do with how Delaware as a state responds. But the question is really centered around how do we interact, how do our federal guidelines and responses for outbreaks conflict or coincide or interact with state or jurisdictional specific responses to conflicts? Is that an issue? Is it still an issue? Is it something we're still contending with? And that could be from the animal or the human side of things.
Sure. I think I can speak to it a little bit from both, maybe, because at UD we work, my department works very closely with a VetLearn perspective in terms of avian influenza and the poultry industry. And then, of course, I've worked with FDA a little bit when it comes to looking at human contamination issues in terms of foodborne illness. So I actually think there is a really nice interaction at the state level and with FDA. And I'll use the produce safety rule as an example. I commend the FDA for the term educate before you regulate. I really think that that's a wonderful sentiment. If there's another aspect that we've learned over the past year is that we cannot let up in terms of the science education that our country needs. We really need to continue to talk to people about how important it is to understand basic biology. And a lot of that is what happens during outbreak investigations. And when we think about disease transmission, I love what Dr. Edwards said earlier about the biosecurity. Just thinking about maybe color coding tools when you're moving from one field to the next, or keeping your truck in, off of one area from another if you have diseased animals there. There are things that, just basic issues about how diseases are transmitted and how we think about surveillance in our own fields. We need to continue to communicate. And I do think, from what I've seen with Delaware Department of Agriculture, that I think FDA is doing a great job. And so I really think it's important that we have all of that multi-pronged approach from the federal and state levels, and we continue to have education as a component of that on all levels. Within our school system, K through 12, it's never too early to really talk about the basics of science. And then in agriculture, teaching our children, wash your hands after you feed your dog. These are all good behaviors to start. And, and we really need to continue to have that discussion about backyard flocks, because they're growing in number and we don't want people to get sick, but we want people to love their animals. And that, I think, is difficult. That might be trickier than the state and federal communication.
Okay. We're just about out of time. I could bring up another subject, although I'm not sure we could cover it in 4 minutes. I'll throw it out there anyway. Antimicrobial resistance. That's a big issue that the agency often puts forward as a big One Health initiative. It's high on the list of WHO, OIE, all of these organizations, because it covers resistance of organisms in animals, in people, in the environment. Just thoughts, random thoughts. Are there gaps? Are there big issues that need to be dealt with there that we could make progress on? Especially, from the federal government to the state, even across the different agencies. And across the different centers of the government. Just throwing that out there. Maybe Dr. Kniel or Dr. Main, you could address that.
Well, I think CVM has done some work here in the use of these antibiotics in growth production in animals, and limiting those and trying to protect the use of the medically important animal antibiotics for human use. So, Dr. Edwards, I don't know if you want to just make a comment or two on some of the efforts CVM has been doing, because that has been one of the big areas of concern, is inappropriate use of antibiotics in animal agriculture. And that is work that FDA has been engaged in and has seen some progress. Dr. Edwards?
Sure thing. Found my unmute button too. So I think as we think about antimicrobial resistance, it is obviously a multi-pronged sort of issue that we have to work on. At Center for Veterinary Medicine, we have worked to ensure that there's the judicious use of those antimicrobials, especially those that are being utilized and are medically important on the human side, because we want to keep those useful for everybody, humans and animals alike. It doesn't help if we wear out antibiotics or have resistance to those by certain pathogens we're trying to take care of. In order to do that, we have worked with some of the sponsors that have come in, and at Center for Veterinary Medicine, as many of you know, we address both animal foods as well as animal drugs. On the animal drug side, we've worked, sponsors have voluntarily pulled the production claims from many of those antimicrobials. I think I mentioned in my talk too that with that change in how some of those antimicrobials are being used, there is additional research and interest in innovating to make sure that animals can stay productive and can continue to grow and produce those products that we eat. And so we have seen a lot of new innovative ingredients that are working in some of the similar spaces, trying to find where we can gain some of those additions to production in order to make sure that we have a healthy and safe food supply, and it's abundant enough for us in the future. So with that change and how a lot of those are utilized, we also have worked to make sure that veterinarians and health professionals are involved in when those antibiotics need to be utilized for treatment of certain diseases. And with that being put in place, that helps us to continue to work towards that One Health goal, to make sure that we have those tools in the toolbox to keep animals and humans safe. So it's a joint effort across many parts of both public and private, and we're excited about what the future brings with that.
Excellent. And with that, I'd love to keep talking, but thanks everybody who's tuned in to watch this this session, and thank you to all of our presenters. It's been an excellent session. I just wanted to thank everybody for attending the 2021 FDA Science Forum, and wanted to emphasize that we hope that you will all visit the 2021 FDA Science Forum virtual poster session exhibition, which is on the forum's webpage at fda.gov. So with that, again, thank you to everybody. It's been very productive. So everybody have a good rest of your day.